Since 2015, George Pernot is Max Planck Fellow and now also Director at Max Planck Institute for Chemical Ecology in Jena, where he has a group on that is focused on the regulation of plankton population dynamics. Now that sounds familiar for many of you. And what he does is he studies chemical interactions between groups for attack and defense, but also for communication, introducing the sexual reproduction between specimens, and so the biochemical signaling. And what is also important, and he doesn't work on that himself, is also that many of these biochemical products, these compounds, are interesting for blue biotechnological applications. But I won't take too much time, so it's up to you, George. You have the floor.
Thank you very much for the introduction, and thanks a lot for inviting me here to give a seminar. It's always a pleasure to be here at the Stazione. It's not the first time, and it's hopefully not the last time that I'm here. So what I want to do today is I want to introduce to you a couple of concepts that we use in chemical ecology to connect chemistry and ecological interactions. And the talk today is divided in two parts. One is providing a view from the big global perspective on the small chemicals that are shuttling around in the plankton. And the other half of the talk, then we go to the perspective of a unicell and see a very local view on the idea of chemicals that are distributed. So first a global and then a local view on the role of chemicals in the plankton and biofilm interactions. So I think there is no better introduction to the system than simply the view on the Earth from space. This is a NASA movie which is very nicely depicting that the chlorophyll in the ocean is really evolving in a very dynamic way over the year. You have massive occurrences of microalgae, blooms of these microalgae, and these form a lot of chemical compounds. Among others, they fix carbon dioxide, and they contribute to carbon dioxide fixation maybe 50% of the global primary production. So every single second breath that we take is due to these microalgae. But this enormous turnover of carbon, of sulfur, of nitrogen is also pumping really the nutrients in the ocean and really contributing to our entire climate functioning. So in a global perspective, we have very relevant players, and these players are unicellular algae. So basically what we look at is a microscopic organism that, by replicating itself a lot of times, is contributing also to global phenomena. And I think I don't have to introduce the system of phytoplankton over here. It's a huge, diverse system. And this diverse system is in really constant, I would say, movement. It's assembling, disassembling, interacting. These guys, they are never alone out there, even if the water is very dilute. And I kind of adapted or borrowed a representation from the Stocker lab in Zurich that illustrates the point that I want to make quite clearly. Basically, every organism, if it's sinking and leaves a trail of chemicals behind, if it's dividing and producing chemicals, if it's sending out pheromone traces, or also if it's dying and releasing chemicals, is contributing to a very complex chemical landscape in the ocean. So it's not only like the NASA map of chlorophyll, but all those key players contribute to a chemical landscape, and this chemical landscape builds up around them. So the projector here is, I think, lacking at least one color, so I have to explain you sometimes what you should see and you don't see. Basically what I wanted to have here in the center is a microalgae that is building up a bloom of chemicals. So this bloom of chemicals is defining the habitat of the algae. So in our perspective as chemical ecologists, the algae is not living in water. but the algae is living in its chemosphere, so in water that is shaped by exuded chemicals. And this is for sure very relevant for the life of the algae in the everyday life, but it's mainly relevant in interactions. So these chemicals that are released into the chemosphere, they can mediate interactions, for example, with competitors. And this will be the topic in the first part of the talk. So this interaction over here can be in a helping way, so they can support each other, but most of the time it's already in an inhibiting way, so they can suppress the competitors. But the interaction can also be with, for example, bacteria, so they can interact, they can feed a specific bacterial community that can support them with, for example, essential nutrients, but they can also fight the bacteria that are not really, I would say, required or maybe even harmful to them. And the algae can interact by chemical means, and this is not a topic of today's talk, but of our research, in terms of chemical defense against grazers like herbivores that are around in the ocean. So basically we have, from the view of a cell, the necessity to produce a large amount of chemicals that mediate all these interactions. And a very important aspect of my talk is that these chemicals, they are not produced all the time. So a lot of the chemicals, being it chemicals from higher plants, from insects, but especially from microorganisms, they are very dynamically regulated. So they are produced upon the interaction situation, they are triggered by chemicals, other chemical stimuli, and then they mediate the interaction. So there is no need to produce an antibiotic if no bacterium is around. There is no need to release an allelopathic compound suppressing the competitors if you're in a comfortable bloom situation and anyway all your neighbors are from your kin. So these regulations, they can be followed up quite nicely, and what we do is we try to bring now the chemistry to a functional way of investigating plankton. So let's start with the idea of chemical interaction of two competing microalgae, or of different competing microalgae, I should say, like Thalassocyra weisflogii and Skeletonema costatum. How would you address that interaction? If you would simply put both into one vessel and observe their growth, you would not be sure that you really observe a chemical interaction. They could have contact interaction, they could shade each other, they take away the nutrient from each other. There are multiple ways how an organism can interact. But what we can do is we can separate those guys with, for example, a membrane that allows free diffusion of the compounds between them. But they are separated in a way that they have no physical contact. They don't shade each other, and they don't interfere by, for example, surface-mediated interactions. And by that, if we now observe the cells in one compartment and the cells in another compartment, we can conclude if indeed the exchange of metabolites between these two compartments over the membrane can mediate the interaction. What we did is we developed these sampling chambers that have also been adapted over here, and I'm quite happy to see that, so that we have sampling pods in there that allow us from time to time to do sampling for the standard parameters like growth parameters, nutrients, and so on, but also, for example, for cell counts or chlorophyll monitoring. And, which is very important for our work, we can scale things up so that we can do also metabolomics over time with the exudates generated. And I want to start with a survey of Lydia Papanikolaou from my lab. I pronounced the name. It's a Greek name, so they are always very long. So Lydia did a survey of chemical interactions of microalgae. So what you observe if you follow the literature on chemical interactions are mainly episodic reports, that algae A suppresses algae B, that algae B suppresses algae C, but there is no system in there. So what we thought is we want to see the frequency and the properties of these interactions. They are called allelopathic interactions. So what we have now in these plots that will follow, a couple of them will follow, are days after inoculation, so 1, 2, 3, 4, 5 days. And we specifically looked only in the first days after inoculation to have cells in a completely nutrient-saturated environment. So we include competition by, for example, nutrients. And the thick green line would be now the control, where we have in both compartments one algae. So what would happen? It should ideally develop as a single culture, and it does, so we have no effect of our culturing vessel. But this is not a result, it's really the basics of the experiment. And now what happens if we substitute in one compartment the algae with another one? And we have now algae that are inhibiting the growth of the competitor. So for example, Prymnesium parvum, it performs best when it's alone. And nearly no matter whom we put aside, is reducing the growth of Prymnesium parvum. So it's one of these algae that performs worse in situations where it's in competition. If you take Dunandiella here, then you have the control bar over here, and you have the other bars over here. What does it mean? Dunandiella always performs better if it has companies. So it's supported universally by the exudates of the others. And then we have algae that show, I would say, diverse patterns. And we have algae that simply don't care. So we have basically every type of interaction in the ocean. Some are supported by the others, some are inhibited by the others, some don't care, and some are very species-specific in their interactions. And in our survey, we were quite surprised to see, which is kind of counterintuitive if you follow the literature, that we have more supportive interaction in the phytoplankton than negative allelopathy, inhibiting interactions. Why is that? Because people tend to report results where one guy is inhibiting the others. Who cares if there's a support? It's really, I think, a bias of human nature that we focus on warfare, on whatever, and we don't see that actually in the ocean there is a lot of helping going on and really support of the others. So we are now wrapping these data together. Lydia is preparing the manuscript, and we want to really, I think, open the view on these interactions, that we should focus really more on helpers. And this is what I want to do now in the next couple of slides. So we have Thalassiosira weissflogii, and this is the algae in our hands that is most universally and most pronouncedly supported by others. So Thalassiosira is a very bad performer if it's alone in the co-culturing chambers. And the cell counts double, triple, pentuple if we have the competitors around in the first days. So it's really, boom, an immediate support of the algae. So what we thought of is there a universal kind of growth factor that is supporting Thalassiosira? And for that, we now need to enter the field of chemical ecology. This was an ecological survey. We established the bioassays. Now we want to look at the chemicals behind that. And we do that by sampling now the water and doing metabolomics. So we need different controls. So, for example, Thalassiosira in both compartments, Skeletonema in both compartments. These would be two controls, the algae alone, and then the interaction situation. And we look now for metabolites that are modulated in the interaction situation, that go down or go up in the interaction situation. We do that by comparative metabolomics. We have a platform in the lab which involves two quite powerful techniques, GCMS and LCMS. We combine those and cover 200 to 450 metabolites in the so-called exometabolome, so what is released from these algae. So we see a couple of compounds that are rather down-regulated or up-regulated in the different situations, and we would be very much interested in these red ones, which are regulated during the interaction. So we do that by multivariate analysis, so we don't compare by hand, but we let the computer do its job. And it's very clear, but also very obvious, that these three treatments are different. The canonical analysis of principal components, it's a multivariate statistic analysis, confirms that. But the more important thing is that the analysis also allows us to ask which compound is regulated. Which compound do we see less or more in the water? So we can generate heat maps of interactions, and in that case, we came up with one compound that was diminished in comparison to the co-culturing situation. And this is the dimethylsulfonopropionate that you see over here. It's not a surprise compound. It's a compound that is known since decades. It is a compound that is very essential in plankton physiology, and I want to explain how it is essential. It is considered as an essential osmoregulator and an antioxidant in phytoplankton. And now, the rationale behind it, what we wanted to ask is, why is there less of the compound in the interaction situation compared to the compound in the skeletonema control? So we derived a chemical analysis for that, and we looked at Thalassocyra and all the other algae that we investigated. What we found is that the dogma that DMSP is a universal osmolyte is nearly true. So basically, all the algae that we investigate, they contain that molecule over here, but not Thalassocyra biesflogii. So it is different compared to the others. It lacks an essential metabolite. So what is the function of that compound? Why is it surprising that an algae lacks an essential metabolite? So let's go to osmoregulation. You all know that the ocean is not homogeneous, but the ocean is patchy. If an algae migrates up and down or with the current, it will encounter different salinities. So, everyday example, a salad leaf. What happens if you place the salad leaf into the salad sauce? It's an experiment. You increase the osmomolarity of the environment, which is standard. What happens if you leave it in too long? It simply shrivels down and shrinks down because it releases water to the environment. The cells shrink and the concentration of salts in the cells thereby increases, and it adapts to the higher salinity. This would be a higher plant. This would be us. This would be many living beings. But now if you consider phytoplankton, it often has very solid, rigid cell walls. Diatoms live in a glass house. They have no flexibility in there. So basically, they have not the option to shrink. What happens now if the salinity increases? It produces osmolytes, and osmolytes are compounds that have a high ionic strength, and DMSP over here has two charges. It's a so-called Zwitter ion with a very high osmotic strength, which is then biosynthesized within the cells. It's a quite simple biosynthesis, it's well understood, and if then the salinity goes down, and this is very important for an osmolyte, it can be degraded again, quite simple, into DMS and an acrylate. We don't know exactly which species it is up to now, and the DMS is then going out into the atmosphere. So a constant assembly and disassembly of that one molecule. Now we come back to the very beginning of the talk, and we look again at the Earth. If you have now the central contributors to, let's say, climate functioning, the central primary producers that all produce that compound over here, it adds up to a petagram scale chemistry. What is petagram? 10 to the power of 15 grams. This is massive. This is really one of the biggest turnover of sulfur that is there globally. And the DMS over here that is released, it's a gaseous compound, it gases out of the ocean. It is the major biological contributor to sulfur in the atmosphere. So we have now a petagram inventory of chemicals that are produced because of these single cellular problems over there. So it's a quite striking and fascinating chemistry that we have. Okay, so what does it mean for Thalassocyra biesflogii? If we now have this compound over here in the surrounding, released by its competitors, it could be utilized by the algae itself, and therefore we need now tools. The tools would be derived by chemical synthesis. So we could synthesize a DMSP that is labeled with deuterium on the methyl groups and with carbon-13 at the carbons over here. This molecule allows us now to monitor the uptake. So we can use mass spectrometry, and what we find is that Thalassocyria westflogii behaves like a sponge for that molecule. Within minutes, it reaches striking amounts of femtomoles per cell of this osmolyte once it is administered to the water. So instead of producing it itself, it has a very efficient uptake mechanism. And we can, by using a lot of mass spectrometrical analysis, I don't go into detail with that, really prove that the entire molecule is taken up. It's not assembled and disassembled during the uptake process, but it has apparently devoted transporters getting in that compound into the cellular context. And this is striking. So what we have now in the plankton is an algae that is universally supported by the others. But how would you term it in an ecological way? It's a prototypic cheater. It cheats its way through life. It sits around, got rid of these genes for biosynthesis of an essential metabolite, but it rather takes it up because the others produce it anyway. So this would be a typical supporting situation in the plankton, so that you need something produced by someone else and then you perform better. And if you shut down the biosynthesis of such an essential and high concentrated metabolite, boom, you can immediately boost your performance. So this would be the situation with the DMSP. So this would be the conventional view at it, and if a compound is there in tera or petagrams, it's usually quite well investigated. And for us as chemists, it was striking that all the data, they are generated really by an indirect method. So for analytics, people add sodium hydroxide to DMSP. It is base-mediated lysis, and they quantify the released gas DMS. It's convenient because you can quantify a gas quite easily. What we now did is and there is now really the color missing. There is a very complex chromatogram behind that. What we did is we developed a method for the direct determination of DMSP. And what we figured out is that DMSP is by no means the only compound around, but that we have an entire complex chemical landscape of related zwitterionic compounds. DMS acetate is one carbon shorter, goniol is a bit longer and has an OH group. So we have a very complex chemistry going on with compounds that are very, very similar in their chemistry. So we asked, what is their usage? What is their, I would say, function in the environment? We don't have one compound, as it was postulated by the indirect measurement, but we have a lot of them. And those would also release, for example, DMS upon base treatment. So it was simply a methodological shortcoming that prevented the in-depth analysis of these central compounds. So Björn in my lab did some experiments where he changed the salinity, and he looked at the concentration of the osmolytes. And what he found is that DMSP, that is considered to be the universal osmolyte, is functioning as salt-dependent osmolyte at the very dramatic changes over here. So it compensates for the massive fluctuations in osmolarity. But the standard situation in plankton is that these organisms will encounter rather minor changes in salinity. And we figured out that, and there is now again something missing, we figured out that the other compounds, like glycine betaine, they compensate the minor fluctuations of osmolarity, as do, for example, DMS acetate, which is over here, or also the others. But there are other compounds which fall into that category, which are not regulated at all. And always in natural sciences, if something behaves differently, it might be the interesting part. So we have a new picture of osmoregulation here, but we also have an entire compound family, like the goniole over here, which is produced by several microalgae. It's not functioning as an osmolyte, even if it has the structural properties. So we reasoned it might be involved in interaction with the bacteria. So it is well known that DMSP is utilized also by marine bacteria and transformed, this would be an arrow here, to DMS in a quantitative way in our experiments. If we now use the shorter chain length compound, it cannot be cleaved anymore, but the methyl is used by the bacteria and methanthiol is released. So bacteria have evolved ways of utilizing these small molecules. If we add mixtures of those, mixtures of gases are released, which is also found in the atmosphere over the ocean. And now we come to the function of these non-regulated compounds. They are always there if a cell is lysed and, for example, the bacteria take their action. And what we found is that goniole, which is not regulated, but structurally related, has a function, surely for the algae, because it inhibits the bacterial metabolism of these metabolites quantitatively. So one of the major carbon sources for bacteria is shut down because of minor amounts of the goniole in the mixture over here. So what we end up, we can extend now our microscopic view that we have compounds that support the cheaters, but also compounds very related to that chemistry that act as inhibitors of bacterial growth. So we have an entire chemical ecology now around this petagram inventory of chemicals. And now I want to connect that inventory with, again, the global chemistry. So we come up with the global sulfur cycle as it is accepted for quite some time. We know that algae and bacteria both produce DMSP, the compound in question. We know that both break it down again to dimethyl sulfide. The dimethyl sulfide is going to the atmosphere, is getting oxidized over there, and as cloud condensation, nucleus is then rained down in form of sulfate into the ocean, and there would be an arrow over here that illustrates that it's then taken up again, and this is the global sulfur cycling. We have then here a very important branching, because the DMS can not only go into the atmosphere, but it's also bacterially oxidized to DMSO and further oxidized to sulfate. So we have a very complex connection over here of 10 to the power of 15 grams of sulfur flying annually around in the ocean. So Kathleen in my lab, and we were very happy to receive the acceptance in Nature 2 weeks ago, thought that she might want to go more into detail with this story. So this was the picture that we drew, and these are all known compounds around. But if you look a bit closer, then you see that within all these phytoplankton cells that we investigated, and we investigated a lot, we found an additional metabolite for a polarity very much related to this DMSP, and it's universally there. What we do is, as chemists, we do structural elucidation often in indirect ways. So first we looked at the mass spectrum of this compound, which was quite surprising. It's heavier than DMSP. It indicates that it still has the sulfur. We know that there are 3 oxygens in the structure, and we came up with a quite strange proposal. This would be an oxidized form of DMSP. Never been reported before, but we needed to verify that, and since we deal with really small amounts, if we think from the perspective of a cellular culture, we derived a synthesis for that. Not trivial, because this sulfoxonium group is highly tricky to get oxidized, but we managed with ruthenium catalysis. It's not relevant over here, and we could show the structure, support the structure, and in co-incubations we could really prove the point that we have found an additional member in the global sulfur cycle. And this member is the DMSO-P. So we have hidden over here DMSO in the molecule, dimethyl sulfoxide, that is, again, in teragrams in the ocean. So what is happening? How is it done that such a compound is really formed? And we cannot rely on the literature existing because there's really only one report on another natural product that has that dimethyl sulfoxonio structural unit. So we have a kind of unique chemistry over here, and we learned in our experiment that it is connected, in fact, to the oxidative and antioxidative cells of condition, let's say it like that, of cells. So if we look at, for example, Isochrysis galbana, and we monitor its growth and we monitor its fluorescence, we see that in terms of oxidative stress, the DMSO-P, so the oxidized version, is upregulated compared to the earlier growth stages. So it is immediately and directly connected to the oxidative stress of cells. So it compensates by obviously mopping up oxidative equivalents that are excess in the stressed cells. But we also can pursue a bit the biosynthesis. To our big surprise, both algae and bacteria produce that DMSO-P, and with bacteria we could also go a bit further into the biosynthesis and found out that apparently DMSP itself is oxidized by a till now unknown oxidase, has to have a tremendous oxidative strength, and that this compound there is formed without demethylation and remethylation. This would have been our favorite pathway in there. Okay, we can also look at the catabolism, and this is now exciting. The catabolism is working in a way very similar to DMSP. So we have now, instead of DMS being released, DMSO being released. And this DMSO is released quantitatively in high amounts by both algae and bacteria. So we have now, if you think of the inventory, the case where we have an additional kind of shortcut in the global sulfur cycle. And we went out by boat to different North Atlantic and Pacific sites. We also did some sampling in the Mediterranean. We found the DMSO-P being present everywhere in picomolar concentrations. That sounds ridiculous, but imagine the amount of water around there. Picomolar concentrations immediately transfer to teragrams in the global scale. So these now kind of take the established sulfur cycle and extend the sulfur cycle with that compound that sits immediately in the center and contributes to the oceanic DMSO, but also explains antioxidative properties of DMSP. So I think for us quite exciting that you can connect a picomolar chemistry with a teragram chemistry simply by playing the numbers and simply by looking into the systems more carefully. So the global perspective of the work was now in there, and now I want to go to a very local perspective of the work. So I told you two parts of the talk. One is the global view and one is the local view. Now we zoom in to the perspective of the algae. So it's all about produced chemicals. I don't stop with that topic here. I don't let you off the hook too easily. But these chemicals are now mediating something else, not global cycling, but very local interactions. With Wim Wiverman, we addressed very nicely, in a very fruitful collaboration, the pheromone chemistry of algae. Some of you are working on pheromones, surely better experts than me as a chemist, but what we did is, together with Wim's group, we looked at Seminavis robusta, which is a diatom that lives in biofilms, as you see over here. And Seminavis has the property that it's capable of moving. It is a diatom, so it's in a silicate shell or cell wall, and it can excrete through slits, so raphe in the cell wall, kind of a slime, and it glides like a slug on slime. And what the diatoms do, you see it over here in a time-lapse video, they glide in a back and forth movement. So they always go in one direction and then they change their direction. Oops. Here, take that cell for example, it goes back and forth, and from time to time it can turn. So basically the movement would be like this and like this with occasional turns. Kind of random, and completely random if no chemical stimulus is around. Okay, now let's go to the physiology of diatoms and to the life cycle of diatoms. Diatoms, as I mentioned, live with a cell wall that is formed from silicate. So it's a rigid glass-like cell wall. And this cell wall is encapsulating the entire cells. If now a cell is dividing, it precipitates silicate, so-called depositioning vesicles, inside the parent cell. So the new cell is formed inside the old cell, and they build up the new cell walls during mitosis in situ inside the protection of the old cell. If you build your new house inside your old house, the consequence of your new house is trivial, it's smaller. Same happens with seminavis. One half is big, the other one is smaller. So if you have repeated mitotic events, cells get smaller and smaller and smaller. And the consequence you see in that picture over here. So here you have cells that are freshly coming from sexual reproduction, they are big, and cells from the same clone, the same strain after multiple mitosis become very small. So if this would go on and on, the cells would simply have no room for their biosynthetic machinery and die, period. But still, we have so many diatoms around, so there has to be a way around that. And this is in form of sexual reproduction. When the cells enter small cell state, they change their properties, the sexual size threshold it is. They change their properties and they develop into mating type. These mating types, plus and minus, would be equivalent to male and female of our sexes. They can mate and form new large cells. They do it by finding each other, by pairing, by removing, shedding away their old and anyway small silicated cells. They have formed gametes, they fuse, form oxospores, and then cells of the initial size that are a lot bigger than these old cells. And then again, mitosis is going on and on and on and on, and boom, a short period of sexual reproduction. So we know that there is some type of synchronization, and people here in the labs work very heavily on that type of synchronization, and we do that as well. And kind of an open question was on the pheromones involved, so the signals that trigger the sexuality and the finding. And what Wim and also we as partners figured out that there are different types of signals involved in the interaction. One signal is released by the cells once they are small, and I call it the I am here signal. So they release a compound that indicates to the world around there is a small diatom cell that is capable of doing sex. I am here, I want to have sex. Okay, simple signal, I think we can all understand that. And then another mating type, the mating type minus, receives that signal and releases an attraction pheromone. It simply says, follow that smell and you will see me, you will not see me, you will find me, we can mate, we can have sex. So if you follow that trace, we can reproduce together sexually. So let's say the I am here signal and the come here signal, the attraction pheromone. And it's not that it hasn't been researched upon, but there were decades of work on these pheromones. It's a very tedious process, and we were very lucky to bring in the concept of comparative metabolomics. Those students that have attended the class before that, you know what I'm talking about, so compare different states of an organism. We can do that very well by investigating the chemical signals from a mating culture, and then we can investigate one of those guys that would immediately release the come here signal, so the attraction pheromone. If we keep them isolated without administering the I am here signal, they are quiet. why should they produce a trace if no partner is around? So they don't call. But if we now take a couple of milliliters from the mating culture and add it here to this empty minus culture, they receive the I am here signal even if the cells are not around. The partners are not there, but the signals are transferred into here. And then they immediately are misguided and produce the follow the trace, the attraction pheromone signal immediately. We can extract those signals on so-called solid phase extraction cartridges, and we figured out that we can pop those guys open and take single beads and ask if they contain the pheromone. Our bioassay looks like that. This would be a polymer bead loaded with the pheromone, and these would be the poor guys that are attracted to the bead and try to mate with a plastic bead. I think it takes no statistics to see that this is a quite effective bioassay. So those guys will never produce offspring. The video is repeated here. It's a 18-minute time lapse, what you can observe. And you see quite clearly an attraction. The way how we evaluate that is that after 10 minutes, we take a photo and we count the cells on the beads. And if we have control beads, they don't attract the mating partners. If we have attractive beads, they really are loaded with the pheromones. So back to our experimental setup, we can also do what chemists like to do, to extract the compounds from the cartridge and to do the statistics or the analytics. We do that by looking at the mass spectra, but in fact, we don't go into detail, but we simply compare and care about the compounds. They should be here. Stupid projector. But we look at those compounds that are upregulated. And in this case, it was really striking because the compound most upregulated upon receiving the I am here signal turned out to be the pheromone. So it was one of those experiments that no one believes you that it's true because it worked. And it worked really from the beginning. So it was striking that people have over decades tried to purify these diatom pheromones, and we come in with a comparative metabolomics, and boom, first signal on our list turned out to be the pheromone. How did we do it? We did some structural elucidation, that's pure chemistry stuff, based on mass spectrometry. Then we synthesized the compound, then we looked at the stereochemistry, we tested, and we found that it is so-called dimer of proline, of L-proline, so it's the diproline as we call it, which is exhibiting the attractive activity. So what we did is then we loaded the pure synthetic compound on our beads and counted the accumulation. And this is a concentration-dependent effect. You have here a logarithmic scale. And this is really the prototypic response of any given receptor-mediated interaction. You have a threshold concentration, which is somewhere here. Then you have 3 orders of magnitude where the pheromone is perfectly active, and nicely enough, it's also the concentration that is occurring in nature. And then you have the higher concentrations where the pheromone is not active anymore. Why would that be? It's an experience that we make all the time. Receptors get saturated. If you are exposed to a smell for a long time, you don't smell it anymore. Someone entering the room would say, "Oh, it stinks here, open the window." We all produce our smell, we don't recognize it anymore. So it's an everyday thing in human life and in diatom life. If the concentration is too high, your receptors are saturated and you don't recognize the signal anymore. So this would be the first search for the pheromone. But we can also use the response of the pheromone, so it's again a time-lapse video, and track the cells and kind of try to understand how they find their partners. So I told you they move back and forward, and I told you that they can accidentally turn over here. And Karen Bondock in my group did a really great work in simulating and investigating diatom movements. What she found is that you have both a so-called chemotactic and a chemokinetic response. Chemokinetic means those guys get faster when the pheromone source is around. Why would they do it? Clearly, if you move faster, the encounter rates are bigger. So they are usually lazy, going back and forth. Nothing is around. If they recognize the I am here, the attraction signal, they speed up and go still back and forth. And they turn. So our first idea was that they maybe turn towards the pheromone. It's not the case. Turning is still random. But what happens is that they have a directional movement by prolonging the forward movement in comparison to the backward movement. So if no pheromone is around, they take the same distance forth and back. If I would be a diatom and over there would be the pheromone source, I would now prolong my forward movement compared to my backward movement. And clear, if I take 3 steps forward and 2 steps backward, I would sooner or later find my mating partner. And this is really what happens in nature. You have that attraction and an orientation, and they speed up, so I would do the same thing running. So quite striking how they find each other. But during these investigations, we also came up with another quite fascinating aspect of the diatom's life. I told you twice already about the very solid cell wall formed of silicate, and to form that cell wall you need dissolved silicate, dissolved silicic acid, to form the biomineralization. And the diatoms are often limited by the availability of silicate. They use the dissolved silicate from the water. You observe often spring blooms of diatoms because the rivers from the coastlines, they bring in a lot of silicate from the stones that are washed out, and then the diatoms bloom. And they take up the silicate, and it's not recycled, but it falls down into the sediments, so it's really taken out of the water. So they require silicate. And we figured out that the diatoms can not only respond to pheromones, but they can also respond to silicate. And it was in the first case an indirect observation by analyzing movement patterns. What we observed in a growing culture, these would be the cell counts, that for sure the silicate as an essential resource is going down. This is trivial. If you have an organism that feeds on something, it goes down. So not a big surprise. But what we also observed, or I should say Karen observed that, is that the movement, the speed of the cells goes up. So if you have starved cells, they are a lot quicker than cells that are fed well. So we verified that hypothesis by doing a series of medium exchange experiments. So we figured out that the cells are quite fast if they have no silicate, that they are quite slow if they have silicate, and if we add now, for example, silicate from the outside world, whoop, they kind of immediately slow down. They are happy and settle down. So we have now quite, I think, interesting idea about the speed, the chemokinesis in response to an external factor, which is not an organic factor. It's an inorganic salt, and there is no prototypic response for that. So what we did, and this is what Karen did, is she did again video monitoring of silicate loaded and controlled beads. I want first to show you the video. It's a time lapse, this is minutes. And then I want to show you the video again and tell you our view on it after we had a half-year time to analyze the movement. So I think even without doing that, you will immediately recognize that the control beads without silicate are not very attractive. And those guys seem to do something. So let's start the video again. So what we observe first is a speeding up of the cells. They go back and forth around the silicate, and they kind of accumulate here. And then the cells get slower and settle down over here in this area. So what happens is that the bead releases a silicate gradient. In that silicate gradient, the cells look for the optimum. And once they found the optimum, they settle down. And the effect becomes slower because after 50 minutes, the silicate is universally distributed due to diffusion and the mixing of the moving cells. So we have a quite clear response that we have again, a chemokinesis and a chemotaxis involved in this search pattern. And this time not against an organic stimulus, as it would be quite established from nature, but against an inorganic salt. We could show that this is really, again, most likely, we're not exactly sure, but most likely a receptor interaction. We are chemists and we love no tool more than our good old periodic table. This is really what we like. So silicium, germanium, these two guys are standing on top, below each other, and we have a chemoattraction to silicic acid, which would be this one here, an accumulation of the cells. If we have no stimulus, that would be the initially red line over here, we find basically no significant response. And if we now add germanic acid, which has kind of similar properties, but the central atom is a bit bigger, it's germanium instead of silicium, then we find that the cells recognize it as well, but they found it to be repellent. So we have an attraction towards the good nutrient and a repellent of germanium, which is also consistent with the fact that germanium is toxic to the diatoms. If you add it in excess, they take it up, put it in their shells, and the shells become porous and crack. So they have not only developed a search behavior for a favorable nutrient, but also an adverse thingy for the toxic nutrients. So they can smell an inorganic salt, strikingly. Okay, last three slides, and then I'll let you off the hook. We can now ask a very important question, and I should ask you the question maybe before lunch: food or sex? So we have now the way of interrogating the diatoms. We can do a behavioral study with unicells because we have two stimuli and can now compare what they do. So what do we know? We know that large Seminavis robusta cells, they respond to silicate. They like the food, but they don't respond to the pheromones because they are above the sexual size threshold. So for the large cells, it's a clear answer: food, not sex. But how about those that have just crossed the sexual size threshold? And how about those that are so small that they need sex to survive? They are just before extinction because they are so small they need the sex. So we can set up an experiment quite easily to test that. We take now the cells that have just become sexual. They are still comfortably big, but small enough to be able to have sex. And we look now at the response. Do you search the pheromone, which is the upper trace here, or do you search the silicate, the nutrient, which is the lower case here? So if the cells are just below that size threshold, they need to have perfect conditions. They have to have enough silicate, so not being starved, and they have to receive the I am here signal to find their mating partner. Otherwise, they don't move. If they are not starved, but they don't receive the I am here signal, they stay where they are. If they are silicate starved, so they have no nutrient but receive the I am here signal, they don't move. Simply no point in moving if you cannot reproduce too well or too efficiently. How about the response to the silicate? Also very consistently, they only respond to the silicate when they are starved. So if you have a cell that just entered the size threshold, it can say, well, food or sex, depending on what is currently my favorite. And if you now have cells that are really small, you can see, boom, 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 the sex is upregulated. They throw overboard all their control mechanisms. They don't care if they receive the I am here signal. They don't care if they are starved. They need the sex. So they select always, in every condition, the sex over the food. Besides really the most adverse conditions, not induced and completely starved, then they stay where they are. But you can see now that even a unicell can have a behavioral ecology, and I think it's quite nice to observe that in diatom we even can do choice assays. So we add to one culture a choice of a pheromone and a silicate-loaded bead. We can see how they attract to the silicate beads when they are not induced and attract to the pheromone beads once they are induced. So with a single signal, we can switch around their preference. Same cells, same experimental setup. In the multiplate, one by one, we can switch their behavioral preference by simply adding a chemical stimulus. Okay, So I hope I could show you that there is a fascination, at least for me, for chemical signals in the environment. I hope I could show you that it's very rewarding to connect them to atmospheric chemistry and to global processes, that everything seems to be connected. And I can thank the entire team in analytical chemistry. That part here would be my group. Then we have Thomas Wichert's group, Chris Steinbeck's group. We are all working on chemical ecology and new concepts, how to approach them. And I can thank you for your attention. Thanks.
Compare the metabolomic profile of different algae in the allelochemical. Did you have also bacteria in that case?
Yes.
Or so it is more complex than when I appeared, but is there any interaction with bacteria in that case with respect to release of the DMSP and the various metabolites?
So we explicitly did not exclude the bacteria because diatoms often perform worse when you have them in an exenic. So most of those interaction partners you could not even grow exenically. So it's often that bacteria contribute to the diatom fitness. And I think it would also be such an artificial situation if you have an organism that is defined by its bacterial flora. It would be like taking a human being and making it sterile. If you don't have your gut bacteria, if you don't have the bacteria on your skin, you would immediately collapse as a human being. So the same is often true for diatoms. They would have an immediate high stress level. So yes, we have the bacteria, and yes, they could modulate. I expect them to modulate, actually. It would be strange if they don't.
Thank you.
Thank you, Gerd. Very interesting talk. I'm going to ask you something about the grazers, even if you didn't discuss this. I don't know if you're working on this as well. And I was wondering about these new metabolites that you found, these DMSO-like metabolites. Mm-hmm. Since the DMS is known to be a feeding attractant, if you have any idea of what if these other molecules do the same, or
it's on the top of the list. So clearly, because up to now, the DMSO was considered to be there. It is known from oceanic chemistry that you have a couple of teratons DMSO floating around. But the concept that it's really there from point sources released by metabolic activity has not been there. So it's the next step we take, that we look at the local gradients, the effect of the gradients. It's a lot of work because you have to establish everything in you. So you have to establish the DMSO analytics in you, you have to find ways how to deliver the compound. And DMSO, even if it's a solvent that we all use in the everyday lab, it's really hard to analyze, and it's really hard to deliver in a clever way, because it has very similar polarities, polarization than water, so we struggle a bit. But I think it's always, if you start a new project, that you struggle first, and we will find a way eventually, hopefully, or someone else. It's going to be out there soon.
Anybody else?
Still one question on the DMSP. This is released naturally by the algae. It's not released after the grazing or after breaking of the cell.
So what you observe is during senescence of a bloom, for example, that you have very high levels of DMSP. And it fits also, I didn't show these results. If we look, for example, at the interaction of Thalassocyra and Skeletonema. If this would be now, let's say, an idealized growth curve of Skeletonema, then we have a consistent support, but we have this type of situation. So the cells leak out DMSP kind of constantly at low level, but then they release a lot of DMSP at the real, I would say, lysis.
One more. So in your allelopathic experiment, when you put together the Thalassiosira westphlogia and Skeletonema, you say that Thalassiosira is not a producer, it's just absorbing the DMSP, which was produced by the Skeletonema.
Yes. Yeah.
Okay. So it was not known before. I mean, you found for the first time that it was producer of DMSP.
So it was known that phytoplankton are universal producer. I think what is striking about the results is that you can identify cells that are not producing. This is the fact, that you have an essential compound that is not produced by a cell, but they rely on the outside world because the DMSP is anyway there all the time. So the production is not so spectacular, but the uptake and the lack of production. This is really interesting. We would be very happy. We haven't started yet. We are not geneticists, to have someone look at the genome of Thalassiosira westphlogiae to see which cell in the DMSP production is knocked out or dead. Because I would assume that it has, at least on a genomic base, a couple of genes still required for the production, but that it's somehow unfunctional. But we would need the excitement of a geneticist. Maybe there is one around here to look at that.
And I guess you tried to look into TAPS, into Thalassiosira pseudonana, for which the genome is available.
It was even, I think, on that. Yes, it has.
Okay.
Mm-hmm. Yeah, it
And
has
you
the
didn't
DMSP
find-
production. Yeah.
Right. Okay.
Yeah, Marina.
I'm so happy that you show how behaviorally complex diatoms are.
Yeah.
Really was very, very interesting. And a comment, because you also have an excellent model organism, not the same, at least for the sexual interaction, the seminavis that can slide and on a substrate. Do you? I mean, I think you also agree that something like that happens in the 3D environment, for the planktonic cells species, and this maybe is also a challenge, how to, let's say, see these interactions for planktonic species.
So I just spent sabbatical time in the lab of Signor Sato, and there we observed really how even planktonic cells, they have these threads that they put out, and then they kind of approach their mating partners. It was very fascinating to see, but it's really the beginning of a story there. We are not yet there. Would be a surprise after a couple of weeks sabbatical, but we start on that. So I think with other planktonic cells it's trickier, because your system, how does it move in a directed way? Maybe, I don't know if you ever tried, it's an adjustment of sinking, because that would be the only movement that I can imagine.
Yes,
That
how can they locate and move?
Locate the mate.
Yeah,
And especially the
in
3D
water
environment of the plankton,
Exactly.
it's highly relevant.
Exactly, that's very difficult to see, no?
Yeah.
Thank you.
But you would need maybe combining the efforts with the Stocker lab that has developed this 3D microscopy to look at the cells. Maybe that would be an entering into the whole story.
Yeah, because we see similar behaviors a bit when you put them on the ground, but it would be nice to see them. Yeah.
I fully agree.
I can imagine that these compounds, the I'm here compound and the follow me compounds, are one of the fastest evolving compounds that there are, because you don't want to signal the wrong diatom. They are sitting there in their biofilms with many different species. Anything known about these compounds in other species?
So we didn't do a systematic review, but what we always do when we have any diatom in the analytics, we always test for the diprolin. So in diatoms we don't find it. It's a quite common compound in bacterial metabolism, and there we focused a lot on the interaction. So for example, we looked how bacteria modulate the pheromone chemistry of diatoms, and we found that certain Rosiobacter species are really degrading the diprolin, and others are really supporting the sexual reproduction of the diatoms. So there is a lot of chemistry going on. There are alternative producers, but in diatoms we didn't find, which is, even if simply that we didn't find it is not a proof that it's not there, but up to now it's not an obvious finding that it's produced by everyone. But we know from the insect world, for example, that there are such confusion strategies that another insect is producing the pheromone from the first one. And so I would be amazed if this would not exist in a biofilm. We simply don't know yet.
If there are no other questions, no, it's time for lunch. But before you all go, there is in the afternoon time for questioning. So if you want to talk with Professor Georg Böhner, please ask me and we find a time slot for you. Thank you very much for having been here, Barbara.